Identifying mutations in monoamine oxidase MAO deficient mice. We are Vincent Huynh, Michael C. Yang, Karen Su. We work under the guidance of Dr. Kevin Chen at the USC Keck School of Medicine.
Some background information. Monoamine oxidase is a neurotransmitter that is responsible for catalyzing the oxidation of monoamines. MAO dysfunction is responsible for a number of neurological disorders. Such disorders include aggression, attention deficit disorder, which is ADD, irregular sexual maturation, and depression.
Some more background information. There are two known types of MAO enzymes. The first type is MAOA, which is an isoenzyme, which is normally found in the mitochondrial outer membrane, the liver, the gastrointestinal tract, and in the placenta of humans. Its function is to deaminate serotonin and norepinephrine. So first of all, serotonin is located in the central nervous system, and it's a neurotransmitter that is involved in inhibiting anger, aggression, mood, and sleep.
Norepinephrine is a neurotransmitter that is also known as noradrenaline. The effects of norepinephrine in human body controls heart rate, blood pressure, and sugar level.
Okay, the second type of enzyme is the isoenzyme MAOB. It is located in the outer mitochondrial membrane. It plays a role in the metabolism of amine groups, and its function is to degrade phenylethylamine. Phenylethylamine is a neurotransmitter that is found in the brain. Its effects in the body includes an increase in blood pressure, glucose level in the blood, and it releases B-endorphin, which is known to cause pleasure. On the left is a picture of MAOA enzyme, and on the right is a picture of MAOB enzyme. The four types of mice we use in our experiment are the wild type, which is a control, MAOA knockout, MAOB knockout, and both AB knockout. One related research is done by Dr. Shi and Dr. Thompson. The project title is Monoamine Oxidase in Neuropsychiatry and Behavior. The second one is done by Anna Scott, Marco Bortolato, Kevin Chen, and Dr. Shi. The project title is Novel Monoamine Oxidase A Knockout Mice with Human-like Spontaneous Mutation.
The purpose of our project is to find ways to identify the genetic mutations that occur within the MAO deficient mice, and also to identify these behavioral irregularities that are caused by these deficiencies.
Hypothesis. So for this project, we have two hypotheses. First, we hypothesize that certain procedures will allow us to identify certain genetic mutation within the mice. And our second hypothesis is that we believe mice with dysfunctional MAOA and MAOB genes are more likely to exhibit neurological disorders than those of wild-type mice. For our project, we have 4 steps of procedures that we use to identify the mutations in our MAO deficient mice. First, we have the genomic DNA isolation, also known as the gDNA isolation. Next, we have the PCR genotyping of gDNA. Third, we have electrophoresis. And fourth, we have a series of behavior tests and scoring.
The first step of our procedure is genomic DNA isolation. In this step, we remove the DNA from tissue samples. The next step we take is PCR genotyping, in which we amplify the DNA that we extracted from the tissue in the PCR machine, and then we take the DNA and run it in the gel electrophoresis. This is an example of gel electrophoresis. We employ an enzyme called Taq polymerase, which is designed to cut the DNA sequence at certain areas. The Taq polymerase is able to cut the normal mice DNA, resulting in the lower band. The mutated DNA would not be able to cut by the Taq polymerase and will result in the higher band, which is seen here. So this process allows us to determine which mice have become mutated.
So in this project, we use a procedure called scoring. Scoring is simply watching a mouse and recording its behavior to in its new environment. For the 6 things we look for in scoring are digging, crossing, rearing, grooming, center, and periphery. So as you can see on the right, we use a behavioral tracker, and we mark the time and the action that each mouse performs. Here is an example of one of our mazes. This particular maze is called the open fields. As you can see, the mouse is placed in a cage where there are no objects, and it is free to roam wherever it chooses. We also divide the cage into a 4x4 grid